Hi, my name is Ariana Baugh, and today I'm going to be sharing one aspect of the research done in the Giudice lab at UNC Chapel Hill, how a process called alternative splicing helps muscle cells grow and function. So, why does this actually matter? I chose this topic because muscle cell health becomes increasingly important as we age. Understanding how muscle cells grow and stay healthy helps scientists study conditions like heart disease and muscle weakness. So, what do muscle cells actually do? Before we talk about genes or RNA, it helps us to remember what muscle cells actually do. They're responsible for everything from walking to heartbeats. They also repair themselves when damaged, which becomes harder as we age. So, the basic biology. Every cell in our body uses DNA as an instruction manual. To use those instructions, the cell makes RNA, and then that RNA helps build proteins. Proteins are what make muscles contract and function. What happens to RNA before it becomes a protein? When a cell first makes RNA, it's not completely ready yet. It's kind of like a rough draft that still needs some editing. One of the most important edits is splicing. This is where the cell removes unnecessary pieces called introns. In the diagram, you can also see that it adds a poly A tail to one end and a guanine cap to the other. What is RNA splicing? Alternative splicing is kind of like editing a video. The cell can cut out different sections of the RNA and then put it together in different ways. This lets one gene make several different versions of a protein. For muscle cells, this flexibility helps them grow, repair damage, and respond to different stressors. You can kind of think of alternative splicing like cooking. Alternative splicing is like having one recipe and then being able to make different versions of the same dish. You start with the same ingredients, but by adding a little something or removing a different item, you can end up with like completely different meals. Cells also do this with RNA. The Giudice Lab's research. The Giudice Lab looks at how muscle cells use alternative splicing while they develop. They study specific genes that help muscle cells organize themselves and move. When splicing goes wrong, it can contribute to different muscle diseases, so the lab pretty much wants to understand how these changes happen. So why do muscle cells use alternative splicing? Muscle cells rely on this because their needs change constantly. Growing, repairing, responding to exercise, these all require different protein versions. Splicing lets cells adapt quickly to their needs. So what happens when splicing goes wrong? When splicing doesn't work correctly, the body might not make the right proteins. This can contribute to muscle weakness, heart problems, and just slower recovery overall after an injury. All of these are issues that become more common with age. The real-world relevance of RNA splicing. This research helps us understand why muscles weaken with age, how certain heart or muscle conditions develop. By understanding how splicing controls muscle health, scientists can find new ways to treat these conditions. The big take-home message. The big idea is that alternative splicing gives muscle cells flexibility by letting one gene make different protein versions. The Giudice lab studies how this process works and what happens when it goes wrong. So, thank you so much for your time. I hope you learned a little something about how RNA splicing impacts the muscles that make up your body.